New topic, related topic, phylogenetics. Phylogenetics, I call this applied evolution. And phylogenetics is the science of crafting those evolutionary trees. You know, we did the taxonomy, we did the classification, right? We put things in groups. But let's put those groups in an evolutionary tree. That's phylogenetics. And so, in evolutionary classification, this is important because we group things according to how we think they're related, and then we base our classification on that. And our nomenclature is based on our classification. So it all starts down with this phylogenetic stuff. So, here's the problem. The methodology is hypothesize a set of relationships. And then group things. Oh, you're all in this, you're all over here in this wing here. We'll put you in this, order. You're over here. We'll put you in this subclass, whatever. We hypothesize relationships, and then we apply the classification. Here's the problem. There was lots of problems. One is, what system are we going to use? How should we craft the phylogeny? By the way, this is called a phylogeny, an evolutionary tree. How do we craft that? What methodology do we use? And then, how do we put the groups on it? And we've been arguing about that for quite a while. I want to give you 3 different ways people have thought about doing this. And then there's the way that almost everyone's doing it today, which I want to explain. And it's important because it's how we define groups. In the last 50, 60 years, there's 3 different schools of thought. And we'll go through them one by one. You don't need to write them down now. But there's these 3 different schools of thought where almost everyone who wants to publish is in this 3rd school of thought here. The first one, the traditional one, would be evolutionary classification. And this was championed by biologists. It's coming out. I mean, some of you took evolution, right? Did you talk about the new synthesis? Does that sound familiar? Well, it's historical. Maybe Amy didn't talk about that. In the early and middle part of the 20th century, evolution was big, and we were classifying, organizing things, and evolutionary classification was the way to go. And it basically said we define the groups, like mammals, and then we name them, we organize them. But the methodology and the philosophy were just what I think is related to one another. As an expert in mammals, this is what I think. So the methodology was not particularly strict, and there was no real underlying philosophy other than relationships, and we based our classification on that. So you get evolutionary trees that kind of look like this. They're kind of fuzzy, and they make you feel good. But they're not real rigorous. That was the challenge. That was the criticism of it. I think a strength of this system was it's based on people who know the stuff best. So a mammalogist working with a particular group of mammals knows them better than anyone else. We should listen to his opinion or her opinion. Other people would say, well, yeah, but that scientist has built-in biases that he or she might not recognize. So the things he or she says might be a little bit skewed by their biases. Sometimes people say you don't want to be biased because science is supposed to be blind, like justice. But on the other hand, sometimes you defer to authority and you listen to people who know what they're talking about. So, two-edged sword, I suppose. So anyway, at the end of the day, this was the problem. Everyone doing it did it a little bit differently. Which again, to me, that's sometimes a strength. Another set that this methodology is pretty much come and gone, and it was really big in the 60s and 70s. It's called numerical taxonomy. You guys take a statistics class? Sokol is the author of the major statistics textbooks. You might have used, I don't know if you used his or not, but this was very mathematically based and called phonetic or numerical taxonomy. And phonetic, think back to your genetics class, phenotype, right? It's what you look like. So this is a type of taxonomy based on similarity. Very old-fashioned in one way, but it's based on similarity on steroids. You gather tons of data, more data than you can analyze, and you run it through computer algorithms. And so you put data on a bunch of organisms into a computer, and the computer uses an algorithm to say, oh, these things are 20% more similar to each other than these things. And so it's based on intentionally analyzed, very statistical similarity. So you say, you can draw a relationship evolutionary tree. These things are, 95% similar to one another. These things are 21% similar to one another. So we rank and classify things based on how similar they are. Now, the people who promoted this were very mathematically oriented. And at one level, they didn't even pretend to think that they were doing evolution. Their mindset, and this is plant people, I think, primarily, their mindset was, evolution is too hard. We'll never understand the relationships, but let's come up with a system that's not going to change much. That's similarity. And they say, and besides, similar things are usually closely related to one another. So if we have enough characters, we have hundreds and hundreds of characters, the things that are most closely related evolutionarily are going to come off similar to one another. And so when we make an evolutionary tree, when we make a tree based on similarity, it's going to be kind of close to evolution, and that's okay. I'm making fun of it because it's not what I do. So on one level, they didn't even care about relatedness. On another level, they said, well, we're approximating it, and we're doing a better job than you can. Because the basic was we can't figure out evolution, so let's not even try. This science only became possible when computers were available, which computers have been around your whole lives, so you don't think back to when they weren't available. But these things ran off huge data sets, these studies. And back in the 60s and well into the 70s, back when I was in high school, that's what a computer program looked like. You had, your computer program was on these punch cards, and that would be a computer program, be 1,000 punch cards that you ran through a punch card reader. Every time you wanted to run the program, you put those cards in there, and they went, and your data, every point of data would be one of these cards punched. Anyone ever seen a punch card? They sell them on eBay now as collectibles. In the mid-70s, this was the most common piece of paper on the planet. But anyway, that's just a little bit of history. So their idea was similarity. And that's okay. That was, it was a way to base fight. And they had a methodology, and they had textbooks on it to show you. There was a textbook on it. But similarity, they would look at it and say that's an extreme example, but it is an extreme example. Here's an example of similarity that would not give us relationships. So here's two sea creatures that have conical shells. If that's the trait you're looking at, you're not looking at another 500 traits. You'd say limpet and a barnacle, and you might put them in the same group. Things with conical shells. Well, limpet, what kind of animal is it? Mollusk, a snail. And a barnacle?
Arthropod.
An arthropod, a crustacean. So barnacles are actually more closely related to crabs evolutionarily. But that one trait you're looking at, you might put them over here. So there are challenges with numerical taxonomy. And we don't do it anymore. The system that's used now, and for about 30 years, is called phylogenetic systematics, or sometimes just called cladistics. It has a very rigorous philosophy underlying it and a very precisely laid out methodology. So if I'm doing the research and you're doing the research, we would come up with similar answers, probably the same answer. So a lot of people like that. I'm a little uncomfortable with that. It's constraining a little bit, but it is a very useful way to study evolution. If you were an evolutionary biologist and you were publishing your research and you were not offering it in a cladistic context, and you sent it to a journal editor and said, I want to publish this evolutionary study of these fishes or insects or whatever, and you were not doing it in a cladistic context, it would be sent right back to you and say, justify why you're not using cladistics. And that's what really got me. That, I think, was kind of authoritarianistic. Anyway, with cladistics, like I said, the underlying philosophy is actually fairly simple and very straightforward and extremely useful. It sounds like I'm not a proponent or a user of this system. I am. I was around when people are being forced into doing it. The older guys were saying, "I don't want to do that," and the younger guys are saying, "Too bad." You draw an evolutionary tree. In a cladistic evolutionary tree, the branches are usually at right angles. That's your clue that you're looking at a cladistically generated one, usually. If they're curvy and bendy, it's not. And we call that a phylogeny. So there it is. The philosophy was first developed by a fellow by the name Willy Hennig. He was a German entomologist, an insect guy. He apparently wrote this textbook, this idea on how to do this in the 50s in German. And only Germans read it, so it didn't go very far. But a few people were seeing it and seeing crude translations of it in the 60s, thinking this is a really powerful tool. And by the early 70s, it was widespread enough. There's a decent translation of it into English, and you know how Americans are, and English people too. It has to be in English, or we're going to ignore it. It took hold, and I think initially in the fish world and later pretty much everywhere. So that's an old-fashioned evolutionary tree. That might be done with evolutionary systematics. That's old-fashioned. It's kind of wavy, kind of unrigid. There's a rigid one. This was made. You don't know how this was made. You do know how this was made, and that's the power of cladistics, the methodology and the consistency. And we call the evolutionary tree. I'm not going to use that word again, but we call it a cladogram. I prefer the word. Where is it? I prefer the word phylogeny. But when you see the right angles, that's a cladogram. Okay? And what Hennig said, what his step, basically, to summarize it down very simply, he said, you group things, let's go to this one here, you define groups based on a unique trait their ancestor evolved. So here's a group, these two types of dinosaurs here. Let's go down and find their ancestor. Here's their ancestor right here. Okay? Maxillary fenestra, extremely complex chambers in the vertebrae, manual digit 4 lost. Okay? Those traits apply to these guys and nobody else. So we hypothesize that trait evolved in their ancestor. And that's what Hennig said. You look for traits, unique traits that define a group. And you hypothesize that trait evolved in the ancestor. Go a little bit further down, you include this lineage here, teeth restricted to front of jaws, larger hands, interlocking caudal vertebrae. That defines all of these things, and nothing down below. And this one group here, Carnosaurs, defined by these traits. These two groups defined by these traits. So in cladistics, you're constantly searching. You're looking at your organisms. You're defining. You're analyzing it. You're gathering data on the characters, and you're looking for characters that only certain ones show. We call those unique features. That's it in a nutshell. This we don't know how this was done. Might have been done along that line, but this has a very rigorous underpinning, and it's now computer algorithms draw these. So in phylogenetic systematics, there's two basic steps. First step, you hypothesize a phylogeny. We call it a cladogram. There's our set of relationships for A, B, C, D, E, F, G, H. And then you rank the groups. And that means you apply the classification, rank the groups. And that's where it gets kind of complicated. And that's why I told you I don't want to spend 3 days talking about this. So you go in here and you say, okay, this is a phylum, or this is a genus, or this is a family. And you apply the classification to your phylogeny. Okay. There you go. Needs expansion. I haven't done that yet. Okay. Did that. Where are we? So let's talk about how we. Let's see. Did that. Yeah. Okay. let's talk about how we make the phylogeny real quickly. You hypothesize a phylogeny. Well, let's figure out how we do it. First, you have to identify your groups, your taxa. They might be species, they might be families, they might be genera. You have to get, what are you working with? They could be phyla. You're making a phylogeny for different phyla. Now, here's an unbreakable rule that cladistics or phylogenetic systematics gives us, and that's all taxa, whether it's a family or a species or a genus, whatever it is, all taxa must be natural. And I defined that word earlier, but here's another word for it. Monophyletic. That word there, break it down. Mono means one. Phyletic means lineage, one lineage. So a natural group is a group that has an ancestor and all the descendant lineages. We don't have any extra lineages in here. We don't exclude any of them. So if it's a family or a genus or a phylum or whatever, it has to be monophyletic. That means the ancestor and everybody that comes off that ancestor. That's why we couldn't put birds in a separate group. Birds are over, reptiles and birds. We had to move birds into reptiles because their ancestor is in there. So if you're not monophyletic, we're not going to give you a family name or a phylum name or whatever. You're not natural. And here's some examples of not being natural. And there's two different ways to not be natural. One is called paraphyletic. We've got monophyletic. We've got three things here. We've got monophyletic, now we have paraphyletic. Para means beside, by the way. And so if you have this group here that's in blue here, and you want to say, let's make this a family. These are species, and let's put them in the same genus. Let's make this a genus. Well, that wouldn't work. If that's the evolutionary tree, that doesn't work because there's the ancestor for some of them at least. But look, we're leaving some out. That's not a monophyletic group. That's a paraphyletic group. If you're leaving somebody out, it's a paraphyletic group. That's reptiles without birds, is paraphyletic. And that doesn't work. Another thing you can't do is polyphyletic. Poly means many or multiple. And here, let's say we have these 5 species, and classification says, let's put them in the same genus for whatever reason we think of. But then you look at the evolutionary tree, and you say we have 2 different lineages here. Polyphyletic. That can't work either. That's not monophyletic. And for the last 40 years, a lot of classification systematics is people going in to groups of insects or fishes or birds and saying, damn, this thing we thought was a nice clean genus, we've got to move this species over here and this species over here. So, did that. Okay. So how do we define natural groups? Well, I've given you a clue on that already. I actually told you. How do we find the natural groups? How do we, these are a bunch of species. How do we define this as a genus? We want to focus here. Now, we don't have this thing. This thing is an ancestor. It's probably extinct. It might have in the fossil record, but you can't count on that. Well, how do we define that thing? How do we find out about that thing? Well, we look at the pieces we do have, and we look for some shared feature. And that shared feature they call a uniquely derived trait, or the fancy biology word is synapomorphy. Apomorphy means, or morphe means feature, syn means shared. So a uniquely derived trait is a synapomorphy. That's the word I use more often. Sometimes they're called apomorphic traits. That's not a word I use, or phrase I use. Let's look at an example. Sorry about allergies today. A synapomorphy is a trait that this guy, see I was going to say invented, that evolved in the ancestor and all the descendants show it. So there's the taxon, Mammalia, class of Mammalia, which we'll get to probably week 4 or 5. What's the synapomorphy?
Mammary glands.
There you go. Mammary glands. All mammals have mammary glands. And that is the trait we use to define the group. And if you go back into time, these are fossil creatures. Go back to the Jurassic, are the first mammals, first fossils we think are mammals. That's where we think mammary glands evolved. And that original mammal radiated in all the things we currently classify as mammals. Right. So what can be a trait? A trait, again, is something that you use to define the group. What can be a trait? Really simple. Anything subject to evolution. Anything subject to evolution can be a trait that you would put on a phylogeny. So, soft anatomy, like muscle structure and soft anatomy, that would work. Hard anatomy works, well, I'd say best, but works really well because fossils leave hard anatomy, so we can incorporate fossils in our studies if we use hard anatomy, like skulls. These are fish skulls. A lot of people poo-poo some other kinds of data and say, well, it doesn't work, but behaviors evolve too. They may be more plastic than bones, but they may not be. But behavior is a trait that's often used in phylogenies. And now we have technologies that we can play with molecules. We can actually sequence individual genes and say A, B, D, D, whatever the genes are, and look for the unique traits there. So anything that's subject to evolution can be incorporated. And this is just me, but a lot of people have lost track of this. Different types of data present different challenges. Different types of data work well in some scenarios and not as well in others. And I think a lot of scientists today have lost track of this concept. So for example, if I'm studying fossil fishes, I'm not going to have in my character list behaviors. It's hard to ascribe a behavior to something we've never seen alive. Sometimes we get clues, but we're not going to do behavior with that. We're not going to get molecules out of that either. You're not going to get a DNA sequence from that creature. Molecules, likewise, aren't much used with fossil things. They're getting molecules out of some pretty old stuff, but not Paleozoic stuff. So if you're doing a study that includes fossil taxa, you're not going to be doing it with molecules. Molecules only work with fairly recent stuff, living stuff. Can't get molecules out of dead things, usually, particularly like something that's recently extinct. This is a fish from Wisconsin. If you're doing a molecular study on that particular group of fishes, you're going to have to leave this guy out because you can't get molecules out of it. Likewise, reserve specimens, historic specimens, not there. Different types of data, different types of challenges. One problem with anatomical data, like bones or even behaviors, is, and we're going to come back and talk about this along the way, is you got a feature like jaws or teeth or something. There is going to be evolutionary signal in there. The teeth evolved from ancestors, so what I call evolutionary signal. But it's also something that the organism uses every day. So there's a lot of form and function, and natural selection may have come at it and altered it around in that particular lineage, and so it looks very different than its ancestors. You may not know that. So sometimes you have to look at anatomy carefully. There's the evolutionary signal, but there's also a lot of form and function, particularly something that's used a lot. So fin supports in fishes, for example, they're very different. We're not going to talk about in this class, we did last spring. But the fin supports, these little structures in the fins, they look the same in a shark or a little bony fish here. They're very, very different. And you need to know that. Sometimes two different things, two unrelated things, are faced with the same niche, same environment, and they acquire a similar adaptation, and they look similar. That's called convergent evolution. Two distant related things getting a similar adaptation. Now, you can't become more closely related, but you can become more similar. And, for example, in Tasmania, we had the Tasmanian wolf, which is a large dog-shaped carnivore, but it was actually a kangaroo-like thing, the Australian carnivore. And very similar in anatomy to a wolf, but very distant related. Two different creatures adapted to the same niche. So that can confuse things if you're playing with anatomy. Molecular people say it doesn't apply to them, but it does. Two very distant related fishes, but they both have the same niche. They eat fish. And that's a really cool tool if you're eating fish. Lots of sharp teeth and a long beak. But you would not put those together near each other in an evolutionary tree based on that trait. Okay. Sometimes creatures are so specialized, they lose a lot of their ancestry. They lose clues to their ancestry. This is an extremely specialized fish called a hagfish, which we'll talk about later. And it's so specialized into its very narrow niche that it's hard to figure out what it is just by looking at it. We often see that in parasites. Parasites can be so incredibly specialized, they lose their identity, at least their visual identity. So hyperspecialization can confuse things. Okay. And this is just me being political, but in the last 30 years, the focus has been on molecular studies and away from actually studying organisms. And that's really, really unfortunate. Non-molecular studies actually encourage you to get to know your organism. I've had molecular biologists in there. You know, can you picture those little plastic tubes you see in lab? That's their only interaction with their organism, is a tiny little piece of frozen tissue in a little tube there. They don't know what the whole thing looks like. And actually working with organisms lets you appreciate and see them. The DNA gel, which is the way that most studies go now. When I was still going scientific meetings in fishes, you used to go and there'd be 4 or 5 days of meetings. There'd be interesting meetings all those days. And by the time last meeting I went to, all the interesting talks were in one afternoon and every other talk was a molecular talk. They jammed everything that wasn't molecular into one afternoon because that was all they were doing then. And you get no insight into behavior, conservation, any other aspect of biology when all you see is that. I think that's really, really unfortunate. But this is the most unfortunate thing. The last 30 years, the focus on molecular studies has meant that lots of other branches of biology have died or are practiced by elderly people who are about to retire. And all we got people doing is that. It became really trendy. And you know why? Because when you're doing this, you've got to buy this stuff, and this stuff is really expensive. And you can go to the university dean, the university president. The university deans and presidents are not extremely bright people. Do not quote me outside the room on that. And to them, money equates significance. And the more expensive something is, the more important it is. But anyway, that's my political point for the day. Okay. So, we define the groups, we find those traits. Whether it's a gene sequence or a particular shape of a bone, we identify the trait, and that allows us to define the groups. So those are synapomorphs. So as we go through vertebrates and we define mammals or amphibians or whatever, the first thing we're going to do when we get to each group is we're going to look at that. We're going to look for that ancestor. Figure out what that ancestor looked like. What unique thing did this guy evolve? Not because he wanted to, just random chance. But what did he get that allowed him to establish a new type of organism? That's the synapomorphy. So back to our example here. The taxon is Mammalia. The synapomorphy is mammary glands. Okay, did that. Let me just check here again. Like I said, let's do this. I'm unsure how summer's going to go, but within a couple of days I'll get it. I'll figure it out. A couple words here. I've been using the word trait or character. Let's narrow them down. Character versus trait. Character is kind of the generic thing. In the case of mammals here, the character is makes milk. Milk production would be the character. But there's two ways to have that character, right? And that's the trait. Yes or no. So in our evolutionary tree, we would code our different organisms. The line in the data sheet would be milk production. And, you'd code yes for makes milk, N for doesn't make milk. So trait is what you are. Character is slightly broader. Maybe another example here. Blood type is the character. You did genetic problems with blood type, right? Anyone find out your father couldn't be your father because of his blood type? I'm always worried that's going to happen in lab one day after people are typing their blood. Okay. Wait, I'm adopted. Blood type is the character, and the traits are A, B, B, and O. The work I did, this is some work I did with African fishes. The character was, there's two bones here, the hyomandibula and the metapterygoid, those two bones right there. The character was, did they touch each other? Did they articulate? And the two traits were no and yes. These two touch, these two don't touch. That's the kind of things you look for. And those are the kind of things that can get coded. So here's the synapomorphic traits, the synapomorphies for vertebrates here. In the blue box here, these are all vertebrates. And there are the traits along there. And we got one more thing we want to look at, and that's how we rank those traits. And there is we can define different groups. In this case here, bass, lizard, horse, monkey. We can define each of our taxa. How do we put them together? And Willie Hennig gave us a method to do that, and he called it the outgroup in-group method. Outgroup and in-group. We've got all our taxa. We've identified all our characters and all our traits. How do we determine what trait means what? And outgroup, in-group. And the in-group is the things you're studying. They're what you're focusing on. And your outgroup is the next thing down the evolutionary tree. The next most primitive thing, as near as you can guess. And in the outgroup, in-group method, what you do is you get all these are your characters, right? With the traits. You also code them for your outgroup. And your outgroup becomes the point of comparison. The outgroup becomes the baseline. And any trait that the outgroup has, you define as primitive, the old-fashioned way to do things. And anything that's in the in-group, that's the more advanced way to do things. And the computer algorithm will come at that. It will use this as a point of reference to say, this is the old way of doing things. This is the baseline. This is this thing here. And the arrangement of characters and traits in your in-group is based on that as the foundation. Again, this is something that would take 3 days to explain. I don't want to go into that, but that's what he gave us. So whenever you're doing a study like this, there's always an outgroup, and that defines the ancient. That defines this thing right here. If you had this thing, that would be ideal. We don't have that thing. We have something that came off of him. So that defines 0, if you want to think of it that way. And this is what we're studying. And the computer algorithm takes the data from your in-group and your out-group, works them around, and draws the trait. And that's a hypothesis. So, quick, this was my doctoral work, which never got published. I drew an evolutionary tree for these African fishes. There's about 30 species, and I drew an evolutionary tree for them. These are all different species. And you can see they're grouped into different higher groups. And my in-group, is in blue here, is all these things. And up there in red, those are the outgroups. Those are what the best people in the business. I went and talked to them. What is the next thing, the closest relative to my guys, what are they? And that was their best guess. So, there's my group of interest. This shows up at PetSmart occasionally. I have some in my apartment. And there's my outgroup. These are the next thing down the evolutionary tree, at least according to Humphrey Greenwood. And so I gathered data from all these different fishes and used these guys, the computer algorithm used these guys to define the base. And then ordered, ranked my species here. So what I already showed you a picture, I used these skeletal features. These are skulls, fish skulls. And I dismantled dozens and dozens of these little fishes and looked at their skulls and compared them. Oh, and gosh, this group of species has this posterior extension for the jugular vein, and this other group doesn't have that. Well, there's a difference. Here's just one example. This is the pelvic girdle of a fish. There's the pelvic fin right there. And in some species, there's called the anterior process of the pelvic girdle is forked. But I had one group of species where it's a little knob. Well, what does it mean? Don't know. Doesn't matter. It's a difference. So the trait, the character is anterior process. What's the structure? The trait is forked or cylindrical. Cool. I had a trait. And there it is right there. The cylindrical one was the weird one, and it's trait 31. And it defines these fishes here. I know you're overwhelmed with interest. So there's the characters and the traits. You know what? Palatine teeth, absent, present. Ventral ossification of the mesethmoid, absent, present. And I had, I don't know, 120, I think, total. These are all my taxa. There's the coding. And it gets put in a computer algorithm, and that's what comes out. Actually, 3 different ones came out. But that is the that's the simplest explanation for these data. That's the set of relationships that explain these data with the fewest branches, the fewest changes. That's a hypothesis right there. And that is a hypothesis on which you can base a classification. You can see, for example, I have 4 different groups of species here. 4 different taxa. I gave each one a genus rank. Before, this was all the same genus, but it appears to be 4 different genera, and actually 2 major groups. So anyway, to sum up, only monophyletic groups can be classified, and monophyly is determined by the presence of unique traits, synapomorphies. Oh, there's a slide that shows that. There's my different unique groups, each one very well defined. This is groundbreaking research. I should have won a Nobel Prize. All right, as we transition towards vertebrates, just a couple more things to do here. See if we can finish this today. Our plan is we'll end when we end. I mean, we'll end by noon, and then on Mondays and Wednesdays we re-adjourn for lab. Is it one, right? Don't have a lot to do in lab today. In fact, since probably a lot of you don't have a lab manual, it'll be homework. But we'll meet over in lab. I think we're in 134. But I got a couple more things I want to do first. Just some practical stuff here that, again, the nuts and bolts here. One of the things Linnaeus gave us, and one of the things he solidified, is the use of the species name. Homo sapiens, for example. And he gave us this, he solidified this idea, he didn't first do it, but he solidified that all species names are binomial. And that means 2 names, 2 words, binomial. So all species names, this is a little fish. This, by the way, this is the evolutionary unit, and I think you never want to lose track of the fact that that's just a symbol for the evolutionary unit. That's the evolutionary unit. That's the thing in the habitat. That's the creature that's evolving. That's just what we call it. So all species names have 2 parts. In this case, this little fish is from swamps in the southeast United States called Atropus maculatus. 2 parts to the name. We call that thing together the species name. That 2-part thing is called the species name. It's got 2 parts. The first word, in this case, Natropus, is the genus. We call it the generic name. Natropus. And the second part, maculatus, is the specific name or the specific epithet. So that second part's not the species name, it's the specific name. I know, it's the distinction with a pretty important difference. Every species on our planet gets a unique combination. You can have a bunch of different species of Natropus. That's a category. You can have, and there's like almost 100 species of Natropus. Notropus maculatus, Notropus atheronoides, it goes on and on and on. But every species gets a unique combination. There can be only one Notropus maculatus, and that's this little guy here. So each species, each individual species gets a specific name, and then that specific name gets assigned to its proper genus. These words have meanings. They translate out of Greek or Latin. Maculatus means spotted. They're usually descriptive. Notropus means keeled back. The original specimens that Jordan had when he described Notropus were dried out, and so this musculature along the back had shriveled, and it looked like it had a sharp keel along the back. That's just an error, but we're stuck with the name. That third part is the person who did the describing of the species. This is how zoologists do it. Botanists do this differently. Zoologists do it this way. Is the person who first came up with the name maculatus gets his name there. That's Hay in 1881. There's the paper. You can look this stuff up. Now notice that Hay's in parentheses. This is where it gets fun. Hay is in parentheses. And what that means to a zoologist is that when Hay first described species maculatus, he put it in a different genus than we have it today. He actually put it in the genus Hemitrema, which is another minnow genus. Subsequently, we rearranged classification, moved maculatus from Hemitrema to Notropus. Hay gets parentheses around his name. I know it's important. When I was publishing a book, here's an example of one of mine, a little fish from Mexico, and my doctoral advisor described that. And when there's no parentheses, that means that the species is still in the same genus as described by the author. So, Mink and I are not wrong. It hasn't been moved. And it won't be, because we're not wrong. Actually, it might get moved to Oligocephalus, but that's okay. Okay. Now, where am I going with this? Yeah. Just real quick, why bother? This is nomenclature. Why bother? Well, this is a native North American fish. We'll get some out in the lab. Because it's a good example of a primitive bony fish. Amia calva is the name. It's a Linnaean name. And whether you're in Russia, China, whatever, it's always Amia calva. I've got books in Chinese. They're all in Chinese, but when you go to the section where they're talking about species, there'll be the Latin script telling you what the name is. So it's always Amia calva, no matter what language you're in. These are what we call vernacular names, or common names for this fish. And the first three lines, just the first three lines alone, are English. It's a North American fish. I've never met anyone called John A. Grendel. That's just an awesome name. Western mudfish, speckled cap. Typically, organisms have a most commonly used common name. This one's bowfin. But there's a whole bunch, and all the different languages there. But it's always Amia calva. So we can always communicate with that. This gets a little bit dicey. So I just like this one. This is a little North American fish. They get about this big. It's called a pirate perch in English. Aphrododotterus cyanus. The species name comes from the guy who collected it, Thomas Say, who was an early naturalist, and it was named after him, Cyanus. And oddly enough, the genus name Aphrododotterus describes where the anus is for some reason. The anus of this fish is a very weird place. It's in the throat. Most fishes have their anus way back here. The intestine snakes all the way up here, and he poops out from his throat. So Aphrododotterus means pooping out your throat. My Greek is not that good. I couldn't come up with a cool name like that, but 200 years ago, people learned Greek and Latin in school. Names can be named after people, to honor people. So, here's a fish from Africa that's named after an important British Museum ichthyologist, Georges Boulanger. This is one of mine, my friend Melanie Stiosny, and she collected those specimens. I named the fish after her. If it's named after a male person, it has an I at the end. If it's named after a female person, it has an A-E at the end. These are Latin. Latin rules apply. Latin is a language with gender, and you Spanish speakers know that. English is a genderless language. Things can be geographic. Whenever you see ensis at the end of a name, it means from, geographically speaking. So this is a little fish from the Congo. Here's Californiensis. Okay. Well, I think this is a good place. Let me just see where we're going here. Probably where I want to get today. Yeah. Let's call it a day here. Give you time to track down lunch if you want. Like I said, I'm still figuring out how summer's going to go with these 3-hour sessions. Let's call it a day here. Is it 1 o'clock or 1:30? I think it's 1, right?
1.
Yeah, so over in the lab at 1, and we'll go down there. So if I have any questions, bring them up. Okay. Yeah. There won't be a